Hello everyone, my name is Renubala. I am a lecturer in biology. My school name is Government Girls Senior Secondary School, Nehru Garden, Jalandhar. I am going to explain the topic mitosis and meiosis, that is the two types of cell division. The topics included in my presentation are: first, I'll talk about the different types of cell division, what is cell division, and the two types of cell division. The first type of cell division is mitosis, and the second type of cell division is meiosis. So, I'll explain the various stages of mitosis first and the significance of mitosis. Then, after that, I'll come to meiosis and the different stages of meiosis. At last, I'll wind up my topic with the difference between mitosis and meiosis. Now let's start with the first topic, what is cell division. First we come to the definition of cell division. Cell division is the process in which one cell, which we call it as parent cell, that gets divided into two daughter cells. This is the normal cell division which occurs in each and every living organism. Now, next we come to what are the different types of cell division, which are mainly of two types. Now, the two types of cell divisions are: first is mitosis, and second one is meiosis. Mitosis is known as equational division. In this, we get two genetically similar cells. We also call them as they are the clones. It is equational division because the chromosome number remains the same as in case of the parent cell. Whereas meiosis, it is a double division. It involves two stages: meiosis 1, which is the reductional division, and meiosis 2, which is the equational division. So the mitosis and meiosis, they differ in case of number of cells also. In mitosis, we get two cells, and in case of meiosis, since it is a double division, so we get four daughter cells. Now, first, I'll explain mitosis and the different stages of mitosis. Then, after that, I'll come to meiosis. Now, first is mitosis, which is the equational division. Let's start with it. Now, what do you mean by mitosis? Mitosis is a type of cell division that results in two identical daughter cells, each with having the same number of chromosomes. Since the number and the kind of chromosomes, they are same, exactly same as their parent cell. So we also call that in mitosis we get a clone of cells, since because they are genetically similar. It occurs in all the growing cells and tissues of the body. It helps in cell repair and general growth of the body. Next comes the stages of mitosis. Mitosis has four stages: prophase, metaphase, anaphase, and telophase. Now, in each and every step of mitosis, in all the four stages, there is a specific kind of event which occurs, and we have to remember that particular event. Now, I will explain all the four stages one by one. Now, we come to the different stages of mitosis. Now, before the first stage, that is the prophase. Prophase is considered to be the first step of mitosis. But before that, the cell undergoes interphase. During interphase, we can see there is a distinct nuclear envelope, nucleolus is there, and the chromatin material is in the form of fine thread like structures. So, in this case, the cell is said to be the active. We consider it to be metabolically active, but otherwise, it is considered to be a resting phase because the cell is not dividing. The cell is preparing itself for the division. Now we come to the first step, that is the prophase. In the prophase, as you can see, the nuclear membrane completely breaks down and the chromosomes, they have become short and thick. They have become condensed. There is condensation of the chromosomal material. So they form compact. The chromosomes, they become short and thick, and the chromosomes are seen to be composed of two chromatids attached together at the centromere. So this is the prophase. Now we come to the second step, that is the metaphase. This phase is considered to be best when anyone wants to study the morphology of chromosomes, then we can study during the metaphase. Why? Because all the chromosomes, they come to align at the equator. They form a metaphasic plate at the center. They align themselves. So this is the best phase when anybody can study the morphology of chromosomes. At this stage, metaphase chromosomes is made up of two sister chromatids which are held together by the centromere. There are two small disc-shaped structures at the surface of centromeres which are known as kinetochores, and these structures are the sites where the spindle fibers, they are attached. So the spindle fibers get attached to kinetochores of the chromosomes. Now the chromosomes, they move to spindle equator. This is the main point which we study during the metaphase. Now next comes the third phase, which is known as the anaphase. Now the most important point during anaphase, that the chromosome, each chromosome has two chromatids attached at the center with the centromere. Now during anaphase, this is the most important step in case of mitosis, because in this the centromere, it splits and the two chromatids, they move towards the opposite poles. Now we consider them as one chromosome is having single chromatid and it is moving towards one pole and other is moving towards the other pole. So this is the reason that, because that the chromosomes number, that remains same. Why? Because centromere split and only the chromatids move towards the opposite poles having its own centromere. So this is the anaphase. Now we come to the fourth step, that is the telophase. Now we come to the final step of karyokinesis during mitosis, that is the division of nucleus. The final step is the telophase. Telophase, at the beginning of this stage, the chromosomes, they have reached to their respective poles. Then they again decondense. You can see there are two nuclei. The chromatids, they again decondense and then they lose their individuality. The individual chromosomes, they cannot be seen now. Now we again see that they see them, the chromatin material, they are in form of a mass at the two poles. So the nuclear envelope, which was previously broken down during the prophase, now it again assembles, it again reforms nucleolus, Golgi complex, and ER, which get disintegrated during the prophase. Now again they reform and become active during the telophase. During telophase, you can see there are two nuclei, but the cell is single. So only the nucleus have divided. The cell have not divided. For that, the cell has to undergo cytokinesis. Now we come to cytokinesis. Cyto means cell, kinesis means division. Cytokinesis is the division of cytoplasm. That is, the cell completely divides into two daughter cells. So, but cytokinesis, the type, it is different in case of animal cell and in case of plant cell. In case of animal cell, cytokinesis occurs by a furrow in the plasma membrane. The furrow starts from outwards and then it extends towards the center, and the cytoplasm divides into two. But in case of plant cell, because there is wall formation, so the cell plate is formed at the center and then it extends outward. So the cell divides. In case of plant cells, the cytokinesis occurs with the help of cell plate, but in case of animals, it occurs with the help of a cleavage furrow. So this completes with mitosis, and at the end we get two daughter cells which are identical to each other as well as to the parent cell. Next we come to the second type of cell division, that is meiosis. It is considered to be the double division because it is grouped under two phases, meiosis 1 and meiosis 2. Each meiosis 1 and 2, each meiosis have the four main phases, prophase, metaphase, anaphase and telophase. These two events, these two divisions, that is meiosis 1 and 2, they are different. The first division is the reductional division, in which the chromosome is reduced to half. But the second division is the equational division. It is similar to mitosis because the chromosome number remains same. So this type of division, at the end we get four haploid cells. So this division occurs during the sexual reproduction while the production of gametes. So we get a haploid set of chromosomes in each gamete so that after fertilization that becomes the diploid cells. So it has a special significance in the sexually reproducing organisms. So the meiosis, it is a type of cell division that results in four daughter cells, each with half the chromosomes of the parent cell. It occurs in the production of gametes in animals. As I have said earlier, that it is of significance in all sexually reproducing organisms because during gamete formation, the chromosome number has to reduce to half because after fertilization, we get a diploid zygote. Now we come to the stages of meiosis. Now, meiosis 1. In meiosis 1, we have four steps, prophase 1, metaphase 1, anaphase 1, and telophase 1. At the end of meiosis 1, we'll get two cells. Then these two cells directly enter into the meiosis 2. Then they will pass through the stages prophase 2, metaphase 2, anaphase 2, telophase 2. And then after that is the last step, that is cytokinesis. And at the end of meiosis, we get four haploid cells. Now I'll discuss each and every step in detail. Prophase 1. Prophase 1 is considered to be a longer and more complex stage of meiosis 1. It is further divided into four substages. First is leptotene, then zygotene, pachytene, diplotene, and diakinesis. During the leptotene, that is the first stage of prophase 1 of meiosis 1, the chromosomes they become visible. The chromosomes they become compact, they become short and thick. Now, the second step is the zygotene. During zygotene, a specific event occurs which is known as synapses. Synapses is the pairing of chromosomes, the homologous pairs. Homologous pairs, they form a pair with the help of a complex structure called synaptonemal complex in between them. So, the complex formed by a pair of synapsed homologous chromosome is called a bivalent. So, this is a bivalent or a tetrad. Why we call it as a tetrad? As you can say, the each chromosome is having two chromatids. So, a pair will have four chromatids. So, it is a tetrad. Now, the third step is the pachytene. Pachytene is considered to be the most important event in case of sexual reproduction because in this the crossing over occurs. Crossing over is the exchange of genetic material between two homologous chromosomes. It is also an enzyme mediated process and the enzyme involved is recombinase. Crossing over leads to recombination of genetic material on the two chromosomes. As you can see here, the one chromosome is shown with the blue color and other with the pink color. And they have crossed over, they have exchanged their corresponding parts. So, one point to be noted here is that crossing over occurs only between the non-sister chromatids of homologous pair of chromosomes. That is the main point, that it always occurs between non-sister chromatids. Now, the fourth step is the diplotene. During the diplotene, the synaptonemal complex, which was formed during zygotene, now it dissolves. But at some points, at the sites of the crossovers, the X-shaped structures you can see. These X-shaped structures are called chiasmata. This one. This is known as the chiasmata, which are showing that the chromosomes or the chromatids they have crossed over. Now, last step is the diakinesis. Diakinesis is it is shown by terminalization of chiasmata. That means the chiasmata also dissolves. Now the chromosomes they separate and the nuclear envelope that also completely breaks down, and at the end of diakinesis the nucleolus disappears and we have the completion of the prophase 1 of meiosis 1. Now we come to metaphase 1. During metaphase 1, all the bivalent chromosomes, as we can say here, we can call them as tetrad also because they are having four chromatids. The bivalent chromosomes they align on the equatorial plate. The microtubules from the opposite poles of the spindle attach to the homologous pair of the chromosomes. So you can see the pair of chromosomes they have aligned themselves at the equator attached at the centromere. Now next comes anaphase 1. Now here is the difference in anaphase 1 of meiosis and anaphase of mitosis. In during mitosis and anaphase of mitosis, the centromere split up. But in case of meiosis. Here, in case of anaphase 1, the centromere do not split and the complete chromosome, along with the two sister chromatids, move towards the opposite poles. So, here is the reason why the chromosome number reduced to half, because the centromere do not split and the complete chromosome with the two sister chromatids move towards the opposite poles. Then, after that, comes the telophase 1. During telophase 1, as we have studied during mitosis, the nuclear membrane and nucleolus they reappear, cytokinesis follows, and then we have two cells. These two cells are known as dyad of cells. Dyad means that there are two cells. Dyad means there are two cells. Two haploid cells are formed at the end of meiosis 1. Now these two haploid cells, they will not undergo cell cycle again. They will not undergo DNA replication again. They will directly enter into the meiosis 2. Meiosis 2 is the equational division. It is similar to that of mitosis. Now we come to meiosis 2. Meiosis 2 also have all the four phases in karyokinesis, that is prophase 2, metaphase 2, anaphase 2 and telophase 2. Now we have these two cells are the haploid cells which we got after meiosis 1. Now these have a single set of chromosomes, that is n number, haploid set of chromosomes. Now these will enter. Again the same thing occurs. The chromosomes they become condensed, nuclear membrane disappears, and the chromosomes again become short and thick. Now they will enter into the metaphase 2. At this stage also, the chromosomes they align at the equator, and the microtubules from opposite sides of the spindle they get attached to the kinetochores of sister chromatids. These are the spindle fibers you can see in the diagram. Now next comes the anaphase 2. Anaphase 2 is similar to the anaphase of mitosis because there is simultaneously splitting of the centromere of each chromosome. What is centromere? It is holding the two sister chromatids together. Now here you can see the two sister chromatids, they are moving towards the opposite poles and the centromere split up into two and each sister chromatid is carrying its own centromere. Now at last we have telophase 2. Telophase 2, the two groups of chromosomes, they get enclosed. They have their own nuclear membrane. They get enclosed by nuclear membrane. Nucleolus again becomes functional and we have in each cell we have two nuclei. So in total how many nuclei we have? Four. Four nuclei and two cells. And then after the cytokinesis we get four haploid cells. Now this ends with the meiosis. At the end of meiosis, one cell will produce four haploid cells. So this is the meiosis. Its significance is that it increases genetic variability. It increases variations. Why? Because crossing over occurs in meiosis 1. So it is of much significance in all the sexually reproducing organisms because crossing over occurs, there is genetic variability. So we are different from each other, from our parents, from our siblings, because meiosis have occurred during the reproduction process. Now, at the end of both the divisions, We have studied mitosis and meiosis both. Now we come to the comparison. Why they are different, mitosis and meiosis. As we have discussed earlier, that during mitosis, the chromosome numbers remain same, although they have the similar steps. Both have both have prophase, but the meiosis, it is a double division. It has two prophase, prophase 1 and then prophase 2. Mitosis, during mitosis, there is equational division. Why equational division? Because the centromere splits and the sister chromatids move towards the opposite poles. But in case of meiosis, there is crossing over in Pachytene stage of prophase 1 of meiosis 1. So there is more chances of introducing variations in meiosis. But in case of mitosis, there is no crossing over, so we get a clone of cells. So the two cells are formed in mitosis, but four haploid cells are formed in meiosis. Mitosis is helpful during the cell growth and repair, but in meiosis is helpful in the sexual reproduction. The gametes are formed which have haploid number of chromosomes and they have half and variations. Variations are introduced during meiosis 1. So, this is the main point which is very significant for the process of evolution also. Now we can see at the end we have two diploid cells after mitosis and four haploid cells are formed after meiosis, each with one set of chromosomes. Haploid means having one set of chromosomes and diploid means having two sets of chromosomes. So these are the differences between mitosis and meiosis. Each division has its own significance. Thank you.